Alright, this is our last lecture. This is RNA viruses and medical importance. Last time we talked about DNA viruses. This time we're going to talk about RNA viruses. And RNA viruses can be classified in a similar mechanism to how the DNA viruses were. We know when we classify a virus, we consider the host range. These are all human viruses. We consider the genome, DNA or RNA, single-stranded, double-stranded, segmented or whole. These are all RNA viruses. We consider whether they are helical or complex or icosahedral. And we consider if they're enveloped or naked. So this is a classification scheme for all of those viruses. Now, we can go through them in this classification scheme, but I tend to not use this as much with the RNA viruses just because there's a lot of overlap. So I'm going to go through and spend a good amount of time on the two RNA viruses that are arguably the most important, at least right now in society. That is influenza and HIV, and then I'll kind of go through a smattering of the other ones at a very surface level. So we'll spend a lot of time on influenza and HIV, and then we'll kind of much later on some of the other viruses. So to start out with, we're going to start with influenza. Influenza is here on this list. It's an orthomyxovirus. So it's a human virus, RNA genome, enveloped, single-stranded RNA, and by the way, you might notice that RNA viruses are almost always single-stranded. They're all single-stranded except for this one group. This one family of reoviruses is the only double-stranded RNA viruses. So again, orthomyxo is a human RNA virus, enveloped, single-stranded, but the thing that's really unusual about the orthomyxoviruses, including influenza, is the fact that it has a segmented genome. In other words, instead of having one long continuous piece of RNA with its genes on there, the orthomyxoviruses have individual segments, usually somewhere between 4 and 8 segments that carry their genome. So specifically here about influenza, influenza is an orthomyxovirus, enveloped, segmented is the really important part, single-stranded RNA genome. You can see a picture here of influenza. You can see it's enveloped. You can see the segmented genome here. In this case, they're showing, what is it, 8 genomic segments. Usually it's between 4 and 8 for a virus to be able to go through its replication cycle. And of course, this is the organism that causes the flu or influenza. We talked about it, excuse me, we talked about a bacteria called Haemophilus influenzae. I told you the story about why they named it that, but it was misnamed because it doesn't cause the flu. The actual causative agent for the flu is this virus that we're talking about now, the influenza virus. All right, so as we look at the structure of influenza, there's a couple of things that really stand out. In fact, there's two really important proteins in their envelope that are extremely important. And those are hemagglutinin, they're showing in purple, and neuraminidase that they're showing in orange. Both of those are in the envelope of influenza. Both of those are absolutely essential for influenza's ability to go through its replication cycle. And the human immune system makes antibodies against both of those as well. So we make antibodies against hemagglutinin and neuraminidase during an infection. So those two proteins are arguably the most important proteins from our perspective on influenza. Let me tell you some more about each of them. First, let's talk about, I'm sorry, my gardeners are out back and making a racket. I hope you guys can hear me okay. First here, hemagglutinin. Hemagglutinin, they're showing in purple. Hemagglutinin is in the viral envelope, and its main job is to attach to host cell receptors. In other words, hemagglutinin is influenza virus's spike. Its whole job is to bind to host cell receptors. So of course, without hemagglutinin, influenza cannot go through its replication cycle. It turns out that one of the genomic segments carries the gene for producing hemagglutinin. You can see that here. Here's the gene to make hemagglutinin. The other really important protein in the envelope here is neuraminidase. Neuraminidase is a protein that's needed in viral budding. So when the virus buds off the host cell, it has to have neuraminidase there. It sort of matures the viruses after they bud. If they don't have neuraminidase, or if neuraminidase isn't active, then the new viruses can't infect the next cell. So it's important for viral maturation is the main job of neuraminidase. And you can see that there is a gene for neuraminidase on one of these segments as well. So again, influenza virus, several different proteins here. The two that are really the most important from our perspective are hemagglutinin, the spike, and neuraminidase, the protein used for maturation of the influenza virus. Both of those, the genes for each of those are on different segments of the influenza virus. And there are different versions of hemagglutinin, and there are different versions of neuraminidase. In fact, we usually describe influenza viruses according to the type of hemagglutinin they have and the type of neuraminidase they have. So when you hear about influenza H1N1, they're talking about the influenza virus that has hemagglutinin type 1 and neuraminidase type 1. When you hear about the influenza virus that's H1N4, it has hemagglutinin type 1 and neuraminidase type 4. Those proteins are so important that we subtype them and we describe the virus according to what kind of hemagglutinin it has and what kind of neuraminidase it has. Generally, that does give you some description of how pathogenic it is as well, because hemagglutinin and neuraminidase are both involved in the replication cycle of influenza. And they're both proteins that we make antibodies against. So it does give you some description of how dangerous the virus is when you talk about hemagglutinin and neuraminidase. All right, so there's the structure. Individual genomic segments, different genes are encoded on different segments. Hemagglutinin is the spike, neuraminidase is the protein for maturation, and we can describe viruses, influenza viruses, according to their type of hemagglutinin and their type of neuraminidase. Well, that segmented genome also complicates the viral life cycle. Remember, this should be a review there of the picture on the right-hand side. This is the viral life cycle. Remember the steps here. The spike on the virus attaches to the host cell receptor. The whole virus penetrates into the host cell. You then have to go through uncoding to release the capsid. The genome is then duplicated. The viral proteins are synthesized. All of that's using host cell enzymes. New viruses are assembled. And then the viruses bud from the host cell to get their envelope. That's the basic cycle for replication of influenza. Well, for any virus, actually. That's the basic replication cycle for any virus. But for influenza, things are a little bit more complex. In this picture, I'm showing them packaging a continuous piece of genome, right, all inside that capsid. But remember, with influenza, there are segments. So during this packaging step, all of those segments have to get inside of the capsid in order for that virus to be infective. So it makes packaging a much bigger deal for influenza. It has to get all of those segments packaged together in order to get a complete version of influenza. I hope I'm being clear here. So packaging is more difficult because you have multiple segments, and all of those segments have to be packaged together into the capsid before you release there. Hopefully I cleared that up. Well, what happens here, imagine if that cell was having a really bad day, and that cell happened to be infected not only by one type of influenza, but maybe two different strains of influenza. Two different influenza viruses are infecting the exact same cell. Well, what could happen is during packaging, little pieces of the genomic segments could get mixed up. You could get some segments from one and some segments from the other packaged together. That happens because influenza has this segmented genome. Because of the segmented genome, it can go through this packaging where you mix up the segments from two different influenza viruses. So if more than one flu virus infects the same cell, the genome segments could get mixed up during packaging. Well, what that means is if you have two flu viruses entering the cell, one is H1N1, the other is H2N2. During packaging, you could get some segments from one virus and some segments from the other virus into the same capsid. And now what you're releasing isn't H1N1 and it isn't H2N2, but instead it's H1N2, a new combination. That's called antigenic shift. Antigenic shift is these rearranging of genomic segments. All right, so influenza has an extremely high mutation rate. Influenza has an incredibly high mutation rate. The genome is constantly changing, and that's because of two different phenomenons. One is antigenic drift, and the other is antigenic shift. Antigenic drift is standard mutations. Remember that mutations are occurring all the time in all organisms on the planet. Viruses also have a mutation rate that occurs. That's called antigenic drift. We are all controlled by antigenic drift. Antigenic drift is constantly going on. It's just the random mutations that occur over time. Now, it is true that viruses have a much higher mutation rate than cellular life forms because they don't have those repair mechanisms for the DNA. So viruses in general have a high antigenic drift. Influenza is no different. Antigenic shift, however, is something else entirely. Antigenic shift occurs because if you have one influenza virus, here the blue one, and that's infecting a cell, and you have here's the red one, and that's infecting a cell, during packaging, you could get pieces of the two merging together to create a new combination. That's called antigenic shift. Antigenic shift is the reassortment of genomic segments. Antigenic shift is the reassortment of genomic segments. Do human beings exhibit antigenic shift? No. Nothing. No other viruses do. It's only seen in segmented viruses. Only segmented viruses exhibit antigenic shift. It's a very specific phenomenon. Influenza is one of the few organisms that exhibits this ability of antigenic shift. And again, it's because it's a segmented genome. And so what I'm telling you is influenza has an incredibly high mutation rate, extremely high, because not only is it mutating like all organisms on the planet, it's also going through antigenic shift and rearranging its genomic segments. Well, unfortunately, every single time you have a new arrangement of influenza, your immune system thinks it's brand new. In other words, immunity to one strain doesn't protect you against other strains. If you are immune to the blue strain and you are immune to the red strain, when the two of them recombine and create one that's part red, part blue, it's brand new to your immune system. You have no protection against the new strain. It turns out that our antibodies attach to both hemagglutinin and neuraminidase together. So our immune system recognizes a combination of H's and N's, not an H and N by itself. So what that means is every time there's a new combination of H's and N's, you wind up with this brand new flu strain. It also turns out that some animals can be infected by some strains of influenza. So for example, in this picture, they're showing you that there are strains of influenza that specifically circulate in bird populations. There are bird strains of influenza. Those bird strains of influenza don't cross into humans very well. They generally don't infect humans at a very high rate. There are other strains of influenza, other H's and N's, that circulate really well in the human population and are human influenza viruses. They don't cross into birds. for some strange reason, it turns out that pigs can be infected both by bird strains and human strains. So the bird strain infects a bird or a pig. The human strain infects a human or a pig. Well, what that means is it makes the pig the perfect antigenic shift environment. If that pig was infected by both the influenza strain from the bird and the influenza strain from the human, inside of the pig it will go through antigenic shift to create a brand new influenza virus. A brand new combination of H's and N's. And of course our immune system, even if it's mostly red, just a small shift here, one genomic segment from the blue, and it's brand new to your immune system. So absolutely you don't have immunity against it. This is why you have to get a flu vaccine every single year. When you get the flu vaccine, you're getting vaccinated against a particular combination of H's and N's. But next year when the flu comes back, without a doubt influenza will have changed through antigenic drift and antigenic shift. So protection against this year's strain will do nothing for you next year. And that's why you got to go through and get your vaccine every single year, because it's a new combination of H's and N's every single year. Okay. So it's also for this reason that living in close contact with animals raises the risk for antigenic shift. We see lots of new strains of influenza coming out of parts of Asia, and that's because people are living in closer contact with animals, both birds and pigs. So it's where you see humans, birds, and pigs living in close contact that you're going to have lots of antigenic shift and new strains of influenza cropping up. That's why we notice that. Now, influenza is more common in the winter. There's been a lot of work done on why is influenza more common in the winter. The really interesting thing is if we look at places in the world where the temperature doesn't change dramatically, there's no association with seasons. Makes us think that it's actually close contact that's the bigger deal. In the winter it's colder, so you stay inside and you breathe recirculated air, and therefore you're more likely to encounter somebody's respiratory droplets and get the flu. It doesn't actually seem to be temperature, it seems to be close contact. Now, the disease influenza here, influenza can cause varying degrees of respiratory illness. Sometimes it's a relatively mild upper respiratory tract infection. In fact, usually it's a relatively mild upper respiratory tract infection. There is variety, however. Different combinations of H's and N's can cause varying degrees of respiratory illness. There have been combinations of H's and N's that cause pneumonia, deadly forms of pneumonia. So it's really dependent. Influenza can be very benign or it can be deadly. It really depends on what particular combination of H's and N's that strain has. If we look back through history, some of the big problems that cropped up with influenza, this is probably one of the worst of all times. In 1918, they called it the Spanish flu, and it was H1N1, hemagglutinin 1 neuraminidase 1. I don't know why they called it the Spanish flu, because it appears that American servicemen during World War I were the major players in transmitting this across the world, but I don't know why the Spanish became so unlucky to have it named after them. But anyway, in 1918, the Spanish flu here, in the 2 years that the Spanish flu was around in the United States, it killed 20 million people worldwide. 20 million people died of the Spanish flu. This is the Spanish flu ward, a gymnasium that's been turned into a Spanish flu ward in 1918. In fact, more people died in the year and a half or 2 years of the Spanish flu epidemic. More people died of Spanish flu than died in World War I plus World War II combined. And that was in the space of about 2 years. So the Spanish flu was horrifically, catastrophically deadly. horrific respiratory tract infections. And in fact, it was really, really strange because usually respiratory infections are most serious for older adults or for young kids. Usually kind of people in their 20s, 30s, 40s are fine with a respiratory tract infection. It's very rarely deadly. But the Spanish flu was an exception. It was bringing down strong, healthy people at an unusually high rate. In fact, it was nearly preferentially killing people that we would usually consider in that healthy group. So that was a particularly terrible combination of H's and N's for the Spanish flu. That was H1N1. Another outbreak, 1968, called the Hong Kong flu. That was H3N2. That had high death rates as well. A couple of years ago now, the bird flu in 2005 was H5N1. That one was extremely deadly, but it wasn't very effectively transmitted from person to person. It was actually originally a bird strain of influenza, and it transmitted from birds into humans, but human-to-human contact didn't seem to transmit it very effectively. So that's the only thing that saved us in 2005. And this past year or two, you've been hearing about swine flu. Swine flu is H1N1. Now, if you remember, Spanish flu was H1N1. This was what set off alarm bells. When epidemiologists realized that what they were seeing a couple of years ago was H1N1, they were crazy, crazy scared, because the last time humankind saw H1N1, it caused the Spanish flu at incredibly high death rates. So as soon as they realized what they were seeing was H1N1, everyone was very scared that we were going to see these horribly high death rates that we saw with Spanish flu. Well, apparently the hemagglutinin and neuraminidase have mutated since we saw it last, because H1N1, while it was more dangerous than typical influenza, nothing like what we saw in 1918. So not nearly as dangerous as what we saw before. Apparently antigenic drift has changed H's and N's since we saw them last. So with influenza, every single year it's different. Every single year we see new combinations. Antigenic shift and antigenic drift are changing those combinations of hemagglutinin and neuraminidase, and it varies on how severe that infection is. Generally, it's most dangerous for young kids and older adults, but there's a lot of variety depending on the combination. Of course, we have no treatments to cure viral infections. We do have some antivirals and, of course, supportive care, like amantadine, Tamiflu, which can slow down the spread of influenza. If we know somebody's infected, we can inhibit its spread through their respiratory tract. We can't cure them, though. Supportive care, you can return fluids, those sorts of things to try and help the patient deal with the effects. Of course, no aspirin. Remember, if you suspect a viral infection, you never give a kid aspirin. If you think they have a viral infection because of Reye's syndrome, that possible complication that can cause encephalitis. And then, of course, best protection we have is the yearly flu vaccine. The yearly flu vaccine will give you protection against whichever strain of influenza they think is coming next. Now, that's a bit of a gamble because remember, influenza is constantly mutating, constantly changing. So when they make the flu vaccine, 6 months before they're going to administer it, when they start to make the flu vaccine, they're kind of putting together their best guess of the H and N combination that's going to infect the United States in the wintertime. How do they do that? All right, surveillance and preparation. So this picture, by the way, used to make much more sense when it was bird flu. Now that it's swine flu, I really need a little pig in the center of that picture instead of the bird, but at any rate. What they do to create these vaccines, they're going to observe the pig, the bird, and the human populations. The CDC is paying attention to what combinations of H's and N's they're seeing in the bird population, wild bird population. They'll check out pig populations, see what H's and N's are seeing there. Human populations, which H's and N's are seeing there. Generally, influenza circles the globe, so from From North America, it moves into Europe. Usually it moves from Asia into North America, so we'll be paying attention to what's happening in Asia, what kind of strains they're seeing there, because generally influenza moves that way across the globe. So the CDC will be paying attention to which strains they see, and in the end they have to make a best guess. Based on all of the evidence that they have, these 3 or 4 or 5 strains are what they think is most likely to show up in the human population in North America when winter comes. Sometimes they make a great guess and they have awesome protection. Sometimes they don't. We never really know what's going to happen until winter comes and the flu hits. And then we know whether or not our vaccine was good. But every year they have to, kind of take their very best stab at creating an effective vaccine. And sometimes they do a great job and sometimes they don't do such a great job. Just differs from year to year. Right, and of course the other thing you can do is try to reduce transmission from birds to humans and from pigs to humans. We know that it moves most effectively from pigs into humans, so trying to limit contact is another way to control the spread of influenza. Now, unfortunately, wild birds carry this, and it's impossible to protect yourself from wild birds, but that's another way to try and limit the spread. And that's what they've done in some parts of Asia, trying to limit contact between, or reduce contact between, especially birds and pigs and young kids, who are generally at the highest risk for influenza. All right, so influenza, that antigenic shift is kind of an unusual idea. At the beginning of lab this week, I will walk you through antigenic shift. We'll kind of draw a picture and I'll remind you of how that works, and hopefully I can clear up any problems you have there. I know it's kind of an odd phenomenon. So I will spend a couple of minutes talking about antigenic shift in influenza at the beginning of lab this week. So if you're feeling a little fuzzy on it, hopefully I can clear it up then. Bring your questions. All right, so we talked about influenza here in the orthomyxo group. The really interesting thing about it is the fact that it is a segmented virus. I now want to talk about HIV, another really big one. I couldn't justify teaching a micro class without talking about HIV, of course. It's here. It's a retrovirus. It is an icosahedral envelope virus with single-stranded RNA genome. The history of HIV here. In the 1980s, there were clusters of very unusual cases of immunodeficiency. Apparently, young, healthy males happened to be homosexual, and this was being identified in larger cities. So in the 1980s, clusters of young, apparently healthy homosexual males were coming down with very unusual infections. The kinds of infections that you do not see in young, healthy people. They would see these very strange immunodeficiency, forms of pneumonia that you don't ever see, forms of cancer that you don't ever see. There was a whole group of these cases. And of course, what's going to happen in that event is the CDC will be notified, and the CDC will realize something is going on, and they'll start to investigate. Eventually, they worked out that they did have an infectious agent. They knew at that point that it was being transferred through body fluids. More data later showed that it could be transferred through lots of different body fluids. And eventually, the Pasteur Institute identified in 1983, actually identified the virus that caused this immune deficiency, and they named it the human immunodeficiency virus, or HIV. And it is the cause for acquired immunodeficiency syndrome, or AIDS. So again, the virus itself is called human immunodeficiency virus, or HIV. The syndrome that it causes is called AIDS. All right, so this virus, we actually know quite a bit about this virus, even though it was only identified in 1983. We know quite a bit about the virus at this point and how it works. It is an enveloped single-stranded RNA virus. It's unusual in the fact that the capsid of HIV does carry a couple of its own enzymes. 3 enzymes that are really quite important to its life cycle. The capsid contains an enzyme called reverse transcriptase. reverse transcriptase makes a DNA copy from the viral RNA. Reverse transcriptase makes a DNA copy from the viral RNA. So remember, the genome of HIV is RNA, and the enzyme reverse transcriptase is actually going to copy that RNA genome into DNA. Turns out that reverse transcriptase has a very high mutation rate, which means it commonly makes mistakes when it makes its DNA, which is part of why HIV will have a very high mutation rate, because reverse transcriptase, the enzyme, has a very high mutation rate. All right, so in the capsid, we have reverse transcriptase that makes a double-stranded DNA copy from the RNA. Another enzyme that HIV contains is called integrase. Integrase does exactly what it sounds like. It integrates the viral DNA into the host cell DNA. So integrase is going to integrate the viral DNA into the host cell DNA. And the other enzyme that's really important for HIV, and he carries it in his capsid, is called protease. Protease is required for viral release and maturation. So as HIV leaves a host cell, protease matures it on its way out. So three enzymes that HIV carries: reverse transcriptase, integrase, and protease. Carries it all in its capsid. Now, of course, these are enzymes that our cells do not have. Our cells don't have reverse transcriptase, we don't have integrase, we don't have protease, so that makes them possible targets. It's a difference between cells that are infected by HIV and cells that are not, because cells infected by HIV would have reverse transcriptase, integrase, protease in them. The other thing about HIV here, HIV has spikes that attach, I really should change that. The spikes on HIV attach to CD4, right? So the receptor on host cells that HIV attaches to is CD4. What kind of cells have CD4? T helper cells. T helper cells have CD4. It turns out that other immune cells do too, but the main ones are T helpers. So in other words, HIV specifically, his spikes specifically attached to CD4 on the T helper. So he's very specifically going to kill off the T helpers. That's kind of a profile of the virus. It has these three enzymes. It attaches to CD4 on T helpers. Now, transmission. How is HIV acquired? It turns out that HIV can be isolated from just about all body secretions. If we look at any body secretion, we can find even trace levels of HIV. Like even things like sweat and tears, we can find trace amounts of HIV. However, only blood, organs, semen, and vaginal fluid have an infectious dose. Remember, the infection doesn't proceed unless you have the infectious dose. Trace levels are not enough. You have to have that infectious dose for the infection to proceed. And only blood, body organs, semen, and vaginal fluid have that infectious dose. So that's the only way to acquire HIV, is through those different methods, right? Or different fluids, I should say. Modes of transmission here, this is statistics for the United States. Most to least common. Male homosexual sex is still the most common transmission route for HIV transmission. 42% of cases are transmitted that way. Heterosexual sex, 33%. IV drug users, 17%. Blood transfusions, transfer from mother to developing fetus, medical and dental procedures, all that's rare. Let me say a word about unknown here. When we say that 8% are unknown, that doesn't mean, holy cow, we have no idea how that person got HIV. That's not what unknown means. Unknown means we could not document it. It could be that the patient was a known IV drug user, but we weren't able to show the link. We weren't able to identify exactly where they got it from. So it has to go into the unknown category. So Unknown doesn't mean, oh no, we have no idea, maybe it was inhalation. That's not what unknown means. Unknown means it was not able to be documented exactly where that infection came from. Almost certainly it's going to fit into one of those high categories. Now I want to make the point that while this is most to least common in the United States, this is not what we see worldwide. Without a doubt, the major mode of transmission worldwide is heterosexual sex, much higher on the list. So heterosexual sex is the most common way to acquire HIV worldwide, but in the U.S., it's still male homosexual sex. All right, so here's another way to look at it. This is transmission in the U.S. between 2001 2004. Transmission rates here. Male homosexual sex is shown in brown. In red is heterosexual sex. Here is IV drug use and then combinations of them. Also, if we look at diagnosis, if we look at the epidemiology, we can consider the age group of the individuals. In this picture, they're showing age group here in green. The main majority of cases, diagnosis occurs between the age of 35 and 49. That's what they're showing in green here. In red, between 20 to 34, in yellow, 50 to 64, and then younger ages and older ages are very rare. That said, we're starting to see diagnosis occurring at a higher rate in seniors and young people. Older and younger people, the numbers are growing. So that's an interesting new development that the CDC is keeping an eye on. Over here on this chart, we're looking at diagnosis per 100,000 people in the United States based on ethnicity. This is all CDC data, of course, trying to keep track of who's acquiring it, how they're acquiring it, when they're acquiring it, so that we can do something about it, so that we can reduce risks, etc. So here we're looking at, according to ethnicity, Caucasians here have a very low transmission risk. African Americans extraordinarily high. Hispanic Americans quite high. Asian Americans a little bit lower. And then Native Americans on par with the Caucasian population. So disproportionately higher in ethnic groups, in African Americans and Hispanic Americans, we see a much higher risk for HIV transmission. We're going to talk more about this in a few moments. So how you acquire it is basically through one of these body fluids, through blood, semen, or vaginal fluids, or transplant of an organ can also transplant it. So once it gets in, this is how HIV enters into the human body. Once it gets in, let's look at what's happening sort of on a cellular scale here. So HIV has entered in. Entry. My cat just completely spazzed out and went running around the corner and skidded out and slid around the corner. Anyway, it's like the floor show while I'm trying to talk to you. If I sound distracted, that's why I'm watching my cat tear up the house. Anyway, all right, so HIV enters into the body. Before our figure starts, HIV goes through this kind of multiplication cycle. It enters into the body, and then it goes through a multiplication step in the dendritic cells and the macrophages. That happens before my picture starts. So HIV enters through sexual contact or IV drug use or organ transplant. HIV enters, the macrophages and the dendritic cells get it, and it goes through a replication cycle in those macrophages or dendritic cells. It then comes out of the macrophages and dendritic cells, and here it is going to attach to CD4 on a T helper. So here you have attachment here to CD4 on a T helper. Next thing that happens is reverse transcriptase. That enzyme, reverse transcriptase, does his job, and he makes DNA from the viral RNA. So here we can see the RNA genome of the virus. Here's reverse transcriptase. It's making a copy of that RNA, creating that double-stranded DNA. The next enzyme is integrase. Remember, integrase is also inside of HIV. Integrase inserts that DNA into the host cell chromosome. So HIV enters, it's caught by the dendritic cells and macrophages, it goes through a replication cycle, explodes out of the dendritic cells and the macrophages, and finds its T helper, attaches to CD4 and the T helper. Reverse transcriptase creates DNA from the viral RNA, integrase inserts it into the host cell chromosome. Now, you have this DNA inserted into the host cell chromosome, and you do have slow, low levels of production of viral genome and protein. So it's sort of this low-level virus replication that's occurring, this slow replication which is releasing new viruses. As those viruses are slowly being released, right, on their way out, they are budding. Protease, the enzyme protease, is used to mature them as they are released from the host cell. And eventually that T helper is killed because it has so many HIV virus particles budding off the surface that it will kill off that host cell. That's what's happening at a cellular level. Let's look at what's happening body-wide here. This is progression of the infection. Several lines on this chart going on all at the same time. I want to pay attention to one at a time. Let's first look at the green trace. The green trace, we are looking at time here on the x-axis. On the y-axis, we're looking at concentration of various things. For the green axis, we're looking at concentration of virus in the blood. All right, so here, you first get infected. HIV first enters in. Remember, it goes through a multiplication cycle in the dendritic cells and macrophages. So you see this great big rise, increase in the number of virus in the bloodstream because it went through replication in the macrophages and dendritic cells. Then the amount of virus in the bloodstream plummets. Why? Because it just entered into the T helpers. It just went into the T helpers. It's hiding inside of the T helpers. You don't see very much in the bloodstream. In fact, it will be a long time period, decades, before you'll start to see higher amounts of HIV exploding out. So very low levels for an extended time period. Now let's look at what would be happening to the T helpers. T helpers are the purple line. So here HIV first infects. You have lots of T helpers. Slowly, HIV starts to kill off the T helpers, so you're seeing kind of a slow decrease in the number of T helpers. The interesting thing is your immune system is amazing. Your immune system will sense the fact that your T helpers are being killed, and it will increase production. So your immune system is going to realize that the T helpers are being killed off, and it will increase production of the T helpers. That's why this line is really not a very sharp decline. It's a very gradual decline because your immune system will go faster and faster and faster and faster in the production of T helpers in an attempt to replace those that it knows are being killed. So as the T helpers are dying off, your immune system is replacing them. But of course, it can't keep up indefinitely. Eventually, it gets to the point where your immune system cannot make T helpers fast enough, and you start to see a sharp drop-off. Right here, the immune system can't make T helpers any faster. And now, as it's killing, as HIV is killing the T helpers, you see a really sharp decline in the a number of T helpers in the bloodstream. Now, if we look at what's happening with antibodies, antibodies is the orange trace. So here, the first time HIV shows up in your bloodstream, takes some time before the antibody level is reliably high enough for us to see. Remember, it takes a while for you to go through and have clonal selection and interaction with the T helper and clonal expansion and plasma cell creation and memory B creation and all that stuff. It takes a while before you'll see an actively high number of antibodies. It can take as much as 6 weeks to get a good antibody production that we can see very clearly. So antibodies don't show up for 6 weeks, can be faster, but 6 weeks is the longest that it can take for antibodies to show up in the bloodstream during an HIV infection. So let's kind of look at what's happening from the patient's perspective here. When the patient first gets infected by HIV, sometimes the patient will exhibit mild symptoms, feeling tired, fluish, achy. Sometimes the patient will report some mild symptoms. If they do, that's probably because of the damage to the macrophages and the dendritic cells that they think is probably what's going on there. But then the patient enters an asymptomatic phase, and that asymptomatic phase on average is 2 to 15 years, but it can be extended much longer. So you have this long time period where the patient shows no symptoms. And the reason the patient is showing no symptoms is because the T helpers are still being produced at a high level. As long as there are sufficient numbers of T helpers, the immune system, by and large, is active. Well, that begs the question, how many T helpers do you have to have for your immune system to function correctly? What's the magic number? It turns out that the magic number is 200 T helper cells per microliter of blood. As long as your immune system has 200 T helpers per microliter or more, your immune system will largely work. If you drop below the 200 mark, the immune system stops working. That seems to be the number of T helpers you've got to have for your immune system to function. So you can be in this long asymptomatic phase where even though the T helpers are slowly being killed off, you still have enough for your immune system to function, and we don't see very many symptoms. It's an asymptomatic phase. Eventually, however, the T helpers will be killed off to the point that the immune system fails. And when you move into that part, we describe that as AIDS. So during the initial infection in this asymptomatic period, we would say that the patient is HIV positive. Patient doesn't become an AIDS patient until they enter into this last section where their immune system is no longer functioning because their T helper levels have fallen below 200. So we call it AIDS when, that should say T helper, when the T helper cell levels fall below 200 per microliter of blood. That's when you're going to start to see symptoms. Because the immune system is no longer functioning, you're going to start to see symptoms, often fever or swollen lymph nodes, diarrhea, weight loss, neurological symptoms, opportunistic infections, cancers, things that crop up because of the lack of immunity. That all will develop here in this last phase, this AIDS phase. So common opportunistic infections, remember opportunistic means that it doesn't infect healthy people, but it can cause damage in immunocompromised people. For AIDS patients, because their immune system is no longer functional because of the loss of T helpers, they're open to lots of different opportunistic infections. Here's some of the most common opportunistic infections. Remember candidiasis. Candidiasis is a yeast infection by Candida albicans of the mouth or vagina. It can cause oral thrush or vaginal candidiasis. Pneumonia. There's lots of different things that can cause pneumonia. One that we see almost exclusively in AIDS patient is pneumocystis carinii pneumonia. It's a very, very rare form of pneumonia caused by a fungus. It's really only seen, or almost only seen, in AIDS patients. So when you see this weird strain of pneumonia, you always test your patient for HIV. Cancers, of course, especially Kaposi sarcoma. I told you about Kaposi sarcoma, that cancer of the endothelial cells, the inner lining of the blood vessels. Extremely rare in a healthy individual. It's almost always seen in AIDS patients now. AIDS wasting syndrome is a really sad complication for reasons that aren't thoroughly understood. When the immune system is not functioning, literally the body starts to kind of degrade its own tissues for nutrient value. Dementia, the immune misfunction can also lead to AIDS dementia. And really other infections. Any infection is much worse if your immune system can't fight it off. So, any infection that wouldn't be very dangerous for other individuals can be catastrophic and deadly for an AIDS patient. Some examples here, some pictures. Here's oral candidiasis. Remember the Candida albicans. This is oral thrush. Here's Kaposi's sarcoma. Remember, Kaposi's sarcoma is a cancer of the endothelial cells, so it really could be anywhere because it's in the blood vessels. We saw Kaposi's sarcoma on the ankle before. Here we're seeing Kaposi's sarcoma in the soft palate inside the mouth, but it really could be anywhere because it's cancer of the endothelial system, of the endothelial cells of the blood vessels. So really rare cancer, again, really only seen at this point in AIDS patients. This is cytomegalovirus causing retinitis. Cytomegalovirus, if you remember, was one of the herpes viruses. It's almost always asymptomatic, except for in immunocompromised individuals. In immunocompromised individuals, it can cause more serious infections, and one of the things it can cause is an eye infection. That's what you're seeing here. This is retinitis in a patient that's gone blind because of cytomegalo infection. Here's AIDS wasting syndrome. We don't thoroughly understand the link, but it appears that when your immune system fails, the body begins to degrade self-tissue for nutrient value. A lot of work is being done on how we can stop AIDS wasting syndrome. It's a really horrific thing to see, this AIDS wasting syndrome, and it does lead to dementia as well, because the patient is degrading neural tissue as well. So questions. I want to go through some questions that I commonly get, and rightfully so, on HIV, because we know so much about HIV. Here's some common questions. First off, where did HIV come from? I know it sort of seems like it came out of nowhere. Well, it turns out that HIV is very closely related to another virus, a virus called the simian immunodeficiency virus. Simian immunodeficiency virus only infects chimpanzees, and HIV only infects humans, but they show a lot of sequence similarity. So what we think happened is that the simian immunodeficiency virus crossed into humans somewhere in the 1930s and 1940s, probably around Zaire. That could have happened through blood contact with a chimpanzee, butchering the animal, skinning the animal, something like that could have easily transmitted the virus from the animal into a human. From that point, HIV does have a high mutation rate because of its reverse transcriptase mutation rate. So the virus then mutated into HIV in humans. We think that at that point HIV was could be found in small villages around that area of the world, and HIV was probably there in small numbers, but it didn't spread very far. It was just seen sort of small levels in these villages. Well, in the 1960s, Africa started to become urbanized. They started to build freeways across the continent. They started to have trade across the continent, trucks that would run from one side to the other. Along with that, things like prostitution became more common. But what they think happened is that actually HIV had been in these small villages probably since the 30s, but in the 60s, when Africa became urbanized, it began to cross the continent. And as it crossed the continent, it then, of course, can spread across the globe. And it continues to mutate even today as it spreads across the globe. So that's probably where HIV came from, is it actually was originally a mutation from the simian immunodeficiency virus. Is there more than one HIV virus? Yes. They have now identified two separate HIV viruses, two separate human immunodeficiency viruses, HIV-1 and HIV-2. HIV-1 is the more common strain. It's the one that you hear about all the time. It is unfortunately also the more pathogenic strain. If somebody is talking about HIV, they're talking about HIV-1. HIV-2 is much less common, much less pathogenic, and we don't know as much about it. So 99% of the time they're talking about HIV-1. If they're talking about HIV-2, they would specifically tell you that they're talking about HIV-2. So HIV-2 is mostly seen in only parts of Africa. It's far less pathogenic. They It appears to be not related to HIV-1 directly, so it appears to be a separate crossover event, a second time that the simian immunodeficiency virus crossed into humans. It doesn't seem to be a mutation of the first HIV, it seems to be a totally separate virus. At this point, again, because the HIV has a high mutation rate, we now see multiple groups of both HIV-1 and HIV-2. We expect to see that because of the mutation rate of HIV. We'll see various different strains or groups. There will always be more variation developing as those viruses continue to mutate and they continue to move. We're going to see more and more variation. The most common strains of HIV-1 in the U.S. are groups E and B. Those are the ones we see most commonly in the U.S. Does everyone infected by HIV develop AIDS? Fascinatingly enough, about 5% of HIV-positive patients never get AIDS. So the answer is no. Not everybody who's infected by HIV develops AIDS. About 5% of HIV-positive patients never develop into AIDS. They are what we call non-progressors. Why this is happening, why they kind of won the lotto here, we don't know. We don't really understand what's happening. Is there a difference in their immune system? Some kind of genetic difference in their immune system is the most likely answer, and so they are looking at that, studying that intensely, looking for possible treatments that could develop from this knowledge. But yeah, about 5% of patients never develop AIDS. Very strange. They're called non-progressors. Magic Johnson, by the way, is a non-progressor. He never has progressed into AIDS. Non-progressors are still infectious, of course, because they are HIV positive. They just haven't developed AIDS. So they are infectious, and the patients and the people that they infect won't be non-progressors. So it's not like it's a strain difference. Instead, it appears to be something inside of their body that's protecting them. Can humans evolve resistance to HIV? There's some evidence to show that yes, we can evolve resistance. Some people remain uninfected even after multiple exposures. There's been some work done with women. There are actually groups of prostitutes in Africa who have been exposed to HIV multiple times. They can document exposures where they've had unprotected sexual contact with somebody who's HIV positive. However, the patients never become HIV positive themselves, which is fascinating. Multiple times they're exposed but never become HIV positive. They remain uninfected after multiple exposures. Why that is possibly some mutation to receptors, including CD4. That's what they're thinking. They did find one mutation for a gene that did protect against HIV exposure. And about 1% of European descendant whites are homozygous for this particular mutation, indicating that they cannot be infected by HIV. So about 1% of European descendant whites. Now, why did they choose that group to work on? Obviously, HIV research should be focused on people of African descent, since Africa has the biggest problem with HIV, but the particular group who worked on this is from Norway. So they were studying, I suppose, people they had easy access to. So they had data on European descendant whites, but really we need to be doing this data on people of African descent so we can identify ways to possibly slow down the spread in Africa. But anyway, so about 1% of European descendant whites have this particular mutation protecting them against infection. They could not be infected. So yes, a lot of work is being done there also to try and understand what's protecting them, and is there any possible way we can use that to vaccinate or protect other people against HIV infection. And of course, yes, theoretically we would evolve this resistance. That's how natural selection works, but of course evolution of the human population would take thousands of years. HIV can mutate much more quickly than we can, so the likelihood of us evolving resistance is not very good. So unfortunately, that's not a very viable option. Question: are these people, these people who have multiple exposures but never get infected, are they contagious for HIV? No, because they never get it. They are exposed, but they never actually become HIV positive, so they are not infectious to other people. Right, testing. How do we test for HIV? The classic way to test is to do a two-part test. The classic way to do this was first to do what's called an ELISA. We talked about this before when we talked about diagnostic tests. The enzyme-linked immunosorbent assay, or the ELISA. The ELISA looks for antibodies against HIV. It is incredibly sensitive. It's also fast and cheap, which is nice. You can run lots and lots of samples very, very quickly, and it's extremely sensitive, so we know we'll find it if it's there. So really fast, cheap, easy to run. The trouble with the ELISA is that it does give a false positive result for 0.2% of patients. In other words, 0.2% of the tests that we run come back positive when the patient does not have HIV. So it's very, very sensitive. Perhaps it's a little bit too sensitive because we do get these false positive results. So the ELISA would be called a screening test, and that's usually how it's used. If you go into a health department and you want to, and they're going to test you for HIV, the first thing that they're going to do is an ELISA. It's a screening test. If you come back negative on the ELISA, they know you don't have it because it's a very sensitive test. So if you come back negative on an ELISA, they'll say, all right, go forth, use a condom, be safe, you're negative. If you come back positive on an ELISA, they want to do the next step, right? They're not sure yet because you could be part of this false positive result. So they got to do the next step. So ELISA is first, it's a screening test. If you come back negative, you don't have it. If you come back positive, they do a Western blot. In a Western blot, they look for not just one antibody against HIV, but they look for multiple antibodies against HIV. It's a very specific test. You do not get false positive results here. It's more expensive, it's more work intensive, it takes longer, so therefore we don't want to do it on every single person, but we use it as a confirmatory test. If you come back positive on the ELISA, then we run a Western blot. And if you come back positive on the Western blot, then yes, you are definitely HIV positive. That's the old school way to do it, is to do the ELISA first as a screening test that's fast and cheap and gets rid of most of the negatives, and then to go forward with the Western blot to confirm the patient's status. They also have newer test kits available, some rapid HIV test kits. They test immediately for antibodies. These test kits have about the same reliability as a Western blot. They're pretty fast. They're very fast and they're very, very reliable. You don't see very many false positive results, but they are quite expensive. But they are available in clinics now, and they are available in hospitals. In all cases, however, I want to remind you that we are talking about testing for antibodies. And whenever we're talking about testing for antibodies, there is the same problem. It takes time for the human body to produce antibodies. And so there's always a 6-week window whenever we're testing for antibodies. It could take up to 6 weeks for your body to produce those antibodies. So you could come back negative even though you're actually positive. And there's been a couple of really sad cases recently with organ transplants. When somebody is an organ donor, we, of course, test them for HIV and hepatitis, among other things. And if they come back negative for HIV, we evaluate their lifestyle to identify if they're at a high risk or a low risk. And if everything checks out, then great, they go ahead and do the transplant. There was a terrible case a couple of years ago where a young, healthy male died in a car accident. They tested him for HIV. He tested negative. He was an organ donor. They evaluated his lifestyle to be low risk for HIV or hepatitis, so they used his organs in transplants. Everything seemed great, and then I think they transplanted, if I remember, his heart, a kidney, and like part of his eye, I think it was. But anyway, a couple of months later, the 3 patients that had gotten organs from him all came down with HIV. Apparently, when he died, he was in that 6-week window where he was infected but didn't have antibodies yet. And that's a really sad scenario. Not only, of course, the young man that died, but also the patients who made it through that transplant operation, and the transplant operation was successful, but now they're dying of HIV, or they are HIV positive. So that is always a problem. When we're testing for antibodies instead of antigen, that is always a problem. It takes time for humans to make antibodies, and so it's not a perfect way to test for sure. Treatments and prevention here. Treatments for HIV. HIV does give us some more treatment possibilities than most viruses do. Because we have those enzymes that are specific to HIV, we have reverse transcriptase, we have protease, we have integrase. Because of that, we do have possible targets. And so there are treatments that target reverse transcriptase. That's what AZT does. There are drugs that target protease, called protease inhibitors. There are drugs that target integrase because of those unusual enzymes. Other drugs, they have drugs that inhibit fusion so that HIV will bind to its receptors but not be able to enter into the host cell. Unfortunately, HIV has an extremely high mutation rate because in part due to the mutation rate of reverse transcriptase. So what that means is that HIV can evolve resistance to the drugs very, very quickly. So what they've started to do is create combinations of drugs, and they call that HAART, Highly Active Antiretroviral Therapy, or HAART. And the idea here is to try and give multiple different drugs all at the same time. So if you're on HAART treatment, they may be giving you a reverse transcriptase inhibitor and a protease inhibitor and an integrase inhibitor and a fusion inhibitor. So you're taking a handful of pills a couple times a day. The idea being, if you give so many different treatments, you're attacking HIV from so many different angles, it cannot mutate fast enough to mutate against all of your drugs at the same time. That's the idea. So you throw all these different drugs at it all at the same time, and even if HIV could mutate against one or two, it can't mutate against all of them. That's the idea there. It's been pretty successful at keeping the mutation rate low by using HAART. Unfortunately, it has some pretty severe side effects. As you can imagine, you're telling patients to take handfuls of drugs multiple times a day, and they're going to have to do this for the rest of their life because there is no cure, of course, for HIV. So this is definitely not a pleasant diagnosis. Some other side effects you can see when patients are on HAART therapy, they'll have some odd side effects, one of which here is the deposit of fat in unusual locations. Here it's on the back of the neck, the fat pad that's developing there. Here it's on the abdomen wall, right? And that's a documented side effect. And of course, patients don't feel well when they're on HAART because they're just simply taking so many medications. It's likely to have pretty severe side effects. Unfortunately, if a patient is on HAART, it's absolutely essential that they take the drugs as prescribed, exactly as prescribed. They cannot miss a dose. They cannot be late with a dose, because if you don't follow the exact regimen, you're raising the risk for mutation. So it's not a pleasant scenario. You give the patient the handful of drugs and you say you have to take it every day, twice a day. I don't care how sick you are, you still have to take it. You can never stop. And you're going to have to do this for the rest of your life. That's the terrible news. Now, the good news is they're getting better and better at altering the HAART therapy. So if a patient is experiencing severe side effects, sometimes they can switch things out. They can take out one reverse transcriptase inhibitor and they can give you an extra protease inhibitor, that sort of thing. So they're getting better at modifying treatment to make it much more feasible for day-to-day life and make quality of life much better for patients. But it's still not a pleasant scenario for sure. So that's inhibiting and slowing down the progression of the virus. Generally, the treatments that we have extend the asymptomatic phase. That's what the best we can do with HIV. Of course, we cannot cure. So our best bet here is to try and prevent. The Holy Grail has been to try and create a vaccine. We would like to create a vaccine to protect patients against original infection. That's the most effective thing we have against other viruses. Unfortunately, HIV is very problematic when it comes to creating a vaccine. No vaccine is close. We don't have any real vaccine on the horizon, so nothing that will be available to the public, at least in the next 5 to 10 years. Research is going on, but it's very difficult with HIV for a couple of reasons. First, HIV only infects humans. Well, if it only infects humans, where are you going to test your vaccine? It makes it very difficult. So a lot of work is done with simian immunodeficiency virus on chimpanzees because it's so similar, but it's not the same thing. HIV infects immune cells. If you're infecting immune cells, those are the exact cells that you want to function during a vaccine. That's what would need to be triggered for your vaccine to work. So we don't even know if it would ever work to create a vaccine against HIV. HIV mutates so quickly. It's entirely possible that we could create this vaccine and 2 years from now the vaccine would be completely useless because HIV would mutate so that the vaccine was no longer useful. And then, of course, HIV can be latent in the chromosomes. So if it's inside of the chromosomes, it's possible that our vaccine would never be effective either. So we don't know if a vaccine is ever going to be effective against HIV. The work is still going on. We know that it's a larger mountain to climb than other vaccines that have been created. So really our best bet still at this point is prevention. Prevention includes, of course, safer sex practices. Using a condom is an excellent way to prevent HIV transmission. In fact, latex condoms and polyurethane condoms do an excellent job of preventing HIV transmission. Natural, the lambskin, all that kind of condoms, those don't do a very good job. They have very large pores in them. But latex condoms or polyurethane condoms do an excellent job of protecting against HIV transmission. Both male and female condoms do a great job of protecting. And I'm talking about condoms that have not been riding around in your wallet since high school. I'm talking about new condoms that are put on correctly and used according to the instructions. They do offer excellent protection. Needle exchange policies and programs have dropped the risk for IV drug users, screening blood products, and organs. We're getting better and better at that, but of course there's always the possibility of somebody being in that window where we don't know they have it. And of course, universal precautions during health care. Again, I told you this at the beginning of the semester, and I'll tell you again, you better wear gloves every time you have contact with a patient. If I get wheeled into the ER and I look up into your face and you're not wearing gloves, you better believe I will lecture you from where I lay in that cot. Right? You better be wearing gloves every time you deal with a patient. And if you have a needle stick, you immediately contact your supervisor, because it turns out that if we put you on some of this heart treatment after a needle stick, the likelihood of getting HIV infection drops significantly. So don't hide that kind of stuff. If you get a needle stick or a tear in the glove or a cut or whatever it is, you got to tell your supervisor immediately, because there are things that can be done to drop the risk factor. So HIV from a global pandemic perspective here, how bad is it? It's really bad. It's really, really bad. About 60 million people worldwide are now HIV positive, 10 million of which are children. 10 million children in this world are infected with HIV. That's mind-boggling and horrifying. More people have died from AIDS than almost any infectious disease ever. If we look at the infectious diseases that kill humans, we look throughout time. The number one killer of humans, the number one infectious disease killer of humans, is smallpox. Smallpox was number one. Killed more humans than any other infectious disease on the planet ever. Number two is AIDS. AIDS has only been around since the 60s, and it's the number two infectious disease killer for mankind. That's a horrifying statistic, and we should all be really, really concerned about HIV and what's going to happen. It's already the number two killer. That's really shocking and horrifying. Number three, by the way, is tuberculosis. So those are the top three infectious disease killers for humans throughout time. The infection rate is still growing. HIV is still a pandemic. Remember, pandemic means that it's increasing in numbers. It is still a pandemic. One would think it would have reached endemic levels at some point, but it has not. It is still and continuing to increase. In fact, we're expecting a big increase. We're starting to see it now. In this decade, we're expecting to see big increases coming from China and India, both countries with large populations that have traditionally seen very, very few cases of HIV. We're starting to see the beginning of an epidemic in both countries. Both countries, again, high populations. We're seeing the beginning of an epidemic. That means that we're probably going to see an explosion of HIV cases in the next 10 years or so. Worldwide, the most common mode of transmission is heterosexual sex, without a doubt. That is definitely the most common. It's not what we see in the United States, but worldwide it's heterosexual sex. In the United States, AIDS is much more common in minorities than it is in the Caucasian population. For example, 40% of all AIDS cases in the United States are in African Americans. But African Americans make up only 13% of our general population. That is a huge disparity. 20% of AIDS cases in the United States are found in Hispanic Americans, while Hispanic Americans make up about 12% of the general population. Again, a huge disparity. 43% of AIDS cases are in Caucasian Americans, while Caucasian Americans make 70% of the general population. In other words, we're seeing a huge disparity between minority Americans, particularly African Americans and Hispanic Americans, in comparison to Caucasians. Now, we can talk about why. There's lots of other epidemiological statistics to indicate why. It turns out that AIDS cases are more common in large cities than they are in rural communities. Well, African American and Hispanic American populations are higher in cities. AIDS cases are more common on the coastline than they are in the center of the United States. And again, African American, Hispanic American populations are higher on the coastline. We can see some correlation to socioeconomic background as well. So there's some evidence to show that there's other things going on besides just ethnic background. But then they're considering other social issues. For example, we know that condom usage and using them correctly is much lower in African Americans than it is in Caucasian Americans. We know that healthcare screening is a problem for both groups as well, or access, I should say, to healthcare screening. So what I'm telling you is there are lots of reasons that have been identified for why AIDS is at such a high rate in minority populations in the U.S. And for that reason, the CDC is focusing all of the money in prevention towards those populations. They feel that if they can target African Americans and Hispanic Americans, we can drop the AIDS cases in the United States dramatically because it is overrepresented in those groups. So they are doing education campaigns and condom distribution and healthcare screenings, etc., in those populations. And that's who they're targeting because they feel like if we can bring these numbers down, we will see a huge drop-off in the AIDS cases in the United States. So that's where the money is being focused. Again, there's only limited resources, so you have to put your money where you can make the biggest impact, and that seems to be the place. And in fact, in the U.S., the fastest growing group of people to get HIV are African American heterosexual women, because heterosexual sex is becoming a bigger and bigger transmission route in the U.S. And so the fastest growing group at the moment is African American heterosexual women. So I wish I had a nicer way to end the HIV portion of this lecture, but I don't. It's horrifying. It's scary. I don't know what's going to happen. But in healthcare, we are coming to a very scary kind of head here where eventually this is really, really going to take over the world. It is slowing a little bit, so that's good, but this is a really scary scenario, and we've kind of become dulled to it because we've heard about it for so long. But again, this is a major killer. Comparing all infectious disease over time, this is a major problem, and we're not very good at tackling it yet. All right, I'm going to come back and do the second part of this lecture. So the second part of this lecture actually will go quite a bit faster. I'm just going to go over a couple of other RNA virus infections, but I'm going to go grab a cup of coffee and then I'll come back.